Which company does produce the LED light?
Semantic. This is screws, right? This might be plants from somatic embryogenesis. This is hybrid aspen.
These are the poplar.
Yeah.
So
Actually, our model species is a hybrid aspen, so a hybrid between the European and the North American aspen. So Populus tremula tremuloides. These are pure aspens, I think. This is greenhouse is more like a backup or when you don't need so controlled conditions.
True.
There are many other grow facilities for more controlled.
wow, it's impressive. So currently, what kind of experiment are you running here?
So, testing various genes, gene combinations that we think will affect growth.
And are you trying to associate phenotypic traits with, for example?
So, this is trying to identify genes that are important for controlling growth, with the idea of developing tools for enhancing growth.
Can you plant GMO varieties in Sweden
Yes.
of trees?
Yes, we have field trials going on in southern Sweden.
Okay.
I've had for 10 years or so these trials almost annually going out in the field.
And how long is the cycle for an experiment of this kind? Like how long is this?
2 to 2.5 months.
2 to 2.5 months. Okay. And can it be longer also?
Well, these trees in 2.5 months, they are 2.5 meters tall.
Yeah.
So that's kind of the maximum
Yeah.
height we can have here. So that's the limiting factor.
Yeah. And how far the solar spectrum resemble the one of the outdoor condition?
If you see now,
Yeah.
these LED lights were supposed to be, they're called daylights.
Yeah.
The lamps. But what we realized when we started doing our experiments here was that the plants got very stunted, short. So we then realized that they were very low infrared light. So then you see we have put up extra far-red lighting here. Now we have a spectra which is very, very similar to daylight. So far-red, red far-red ratio about 1.5 or something like that.
So, because we are struggling a lot in our project to get close to something that is sufficiently similar.
Yeah.
How do you have the UV component?
There is a UV component in this light, but I cannot tell you how similar it is to daylight.
Yeah. Indeed, it's very difficult to quantify because it's like a very complex spectrum.
We have been much more focusing on the red, far red end of the spectrum.
Yeah, because probably that is definitely one of the most important for the morphology.
If you just look at, if we just take out the light spectra, you will see that it's very similar to daylight, except for on the far red end of the spectrum.
Yeah.
So that's why we added this spectrum.
And in terms of uniformity, did you already make some check to see how uniform is the light intensity over the surface?
Well, it doesn't matter so much when you have a system like this because the plants move all the time, right?
Yeah.
We have relatively high uniformity. I mean, if you look on the wall there, you can see this.
Yeah, indeed, I was.
Light is spreading now.
Yeah.
So you see that what we actually do is that we raise the lamps as the trees grow.
Yeah.
So we can raise them up. And that's in order to keep the trees within the uniform part. Otherwise, you see that if they were been growing between the rows here, they would be rather dark after a while.
Yes,
so
yes.
Therefore, we raise the lamps, but then as all the trees move in this direction, they will change places all the time. So that means that on average, you get extremely uniform.
Yeah. And so because the aim of a facility like this is to get as close as possible to outdoor condition. Yeah. And I wonder, please correct me, yeah.
This is a tricky one, huh? I mean, you can really debate that a lot on you always have to make this trade-off between being as close to natural conditions as possible and having as reproducible conditions as possible. I would say that to get very, very close to outdoor conditions, it's better to be in a greenhouse, no? And that when it's actually the natural light which is the majority, because then you will also get the differences between morning and evening and all these changes in light composition which occurs over the day. Yeah. On the other hand, you will get very different growth conditions in the summer and in the winter and so on. So you have to make a trade-off there.
Yeah, exactly. Because the goal of our project is to be able to mimic a specific location as close as possible. Because we hope that in this way we could have like some information about how the yield and the phenotyping more in general will be. So how in the end we want to build suitability maps around the world for the crop of interest, hazelnut in this case. So indeed I agree indeed like that is we have to make a system that is able to reproduce the experiment in order to avoid artifacts and things like that. But at the same time, indeed, we want to do this thing that is a little bit utopian, because we will never be able to match completely the outdoor environment. It's like it's just a vision. We want to go in that direction, to the point in which we will have a proxy of the outdoor conditions. And for example, a question we are struggling around, in your knowledge, how different is having just top light or having like a light system that follows the natural change in inclination?
I don't know if that has ever been really studied. I mean, I think what's much more important than the angle of the light is the light quality. I mean, you have LED, full spectrum LED lamps where you can simulate different light qualities in the morning and the evening. No?
Mm-hmm.
So because that's what's happening. In the morning you get much more far red light. And then the light spectra changes, so you get in the middle of the day you have a red far red light ratio of about 1, and then it drops again in the evening. And this is probably very, very important for but in normal greenhouse conditions you don't care about that. But if you have a lot of outside light in your greenhouses, then you will get these light composition changes also. In here you will not. We can, if we want to, give them far red only at the end of day or end of night. We kind of simulate this. We haven't started to play around with this yet. I mean, there are LED lamps. Baloya, for instance, have these lamps, I know, where you can program them to simulate the light conditions at a specific place on Earth.
Yeah.
At a specific latitude at a specific time of the year.
Yeah.
So they change the light composition over the day. If that is what you're interested in. They're quite expensive, but that's possible.
And in terms of light intensity, because that's another variable that is often difficult to push to the best.
Yeah. You can never get like a summer day intensities in a setup like this. That's like a really bright summer day is like 2,000 micro-Einsteins or something like that.
Yeah.
That's really a lot of energy. It would require extremely strong ventilation systems to be able to maintain temperatures. Also lamps that will, I don't think any LED lamps can achieve that, so we need a lot of really strong.
And there is a topic that we have been struggling a lot conceptually recently. That is about experimental design, specifically about what to consider an experimental unit. So, because I know that when you are doing these studies of, for example, phenotyping on from a genetic point of view, like trying to associate genes to a specific phenotype, and you have different varieties in the same experiment, you consider the single plant as an experimental unit, if I'm not wrong.
Yes.
Yeah. But when we do experiment at the climate level, for example, what we do often in Wageningen, so we are interested in like the effect of a factor that can be light. We consider the whole plot. For example, this chamber will be a single experimental unit.
Well, I mean, if you have a uniform genotype, so you have a clone that you're investigating, then that is the experimental unit for sure. Huh?
Yeah. If you want to use this facility indeed to study, for example, the effect of end-of-day far red, let's say, on one clone, you run this system for 2 months, and for the 300 plants that are in, and that's one experimental unit. So is that the only way to use this facility? Because it starts to be very expensive. If the aim indeed is the one that I said.
But I mean, what is it you want to do? You want to compare different light treatments.
For example, yeah.
So then you need to be able to divide the facility in having certain light treatment at one end and certain light treatment at the other end.
Yeah, let's say
that would be kind of difficult.
Yeah, but let's say that we do that. For example, that we have a screen between.
Yeah, well, then you have. Then of course you can run 2 different conditions at once. Yes.
And in that case, will you feel comfortable in using a single plant as like an experimental unit, or that's the whole compartment that will be according to the
whole compartment? If it's a clone, the whole compartment will be one unit.
Yeah. And are you planning to do such experiment in this facility?
No, we cannot shield off here.
Okay. But let's.
So if we want to do that, we first run one experiment with one light condition and then another experiment with another light condition.
Yeah. But then that's experimental unit 1, so you have to do at least the same experiment 4 or 5 times. So it means that the whole thing will take 2 years.
I mean, I guess that depends on how convinced you are about the reproducibility of your conditions.
Yeah.
I mean, here we have extremely high, very, very little variation. I mean, if you did that in a greenhouse,
Yeah.
absolutely, you would have to do it because it would be almost impossible
Yeah.
to repeat the experimental, the growth conditions completely the same. But here you can, there's very little variation.
Yeah. So, because indeed, what often I see in certain publication that is that they use like the plants that are actually pseudo replicas as replicas, and they do the statistical analysis. Of course, the quality of the research is a bit less if you don't have a highly reproducible facility. But indeed, I wonder if yeah. So when you have a facility of this kind, it's possible to feel sufficiently confident in assuming that this is highly reproducible.
I mean, I think we would very rarely do our experiments like this, right? Because we have a uniform growth setup here.
Mm-hmm.
Then what we want to do is investigate the function of genes under a very particular homogeneous growth conditions. We don't want to change the growth conditions. But we don't want to introduce that variable. We want to know what is the genetic component here responsible for certain changes in growth rates or growth quality.
Yeah.
So then we don't change the growth conditions. They are standardized. So then in a single experiment like this, we have lots of replicates, we have lots of different genotypes. So then we have all our, lots of experimental units here.
And indeed, what would you advise to us? That our goal is indeed like to be able to have like a test of performance of the plant in a specific climate. How could we get like significant results, statistically significant results? Because you can imagine that also, as we discussed yesterday, like hazelnut has a long reproductive cycle.
Yeah, I mean, maybe you have to build a facility with lots of compartments. That's not so easy. This is in a way similar to the question about, how would you perform a field trial?
Yeah.
To see how different clones are adapting to different climatic conditions. And in a normal field trial, you would not replicate the field trial in the same place after each other, right? Because it takes too long.
Yeah.
You don't want to do that. So typically what you do is that you have an experimental design where you say, okay, these are the conditions for this particular plot that we compare to the conditions of a different experimental plots. And then you have your random design with clones and all this. So you will not replicate the field trials in that sense. You could do it in very, very large experimental setups. So for instance, when we test progeny from process of trees in Sweden, we plant them at several different locations in Sweden at the same time, clonal propagated material. Then you increase the power a bit, of course, but otherwise I think you just have to, you can divide an experiment like this in sub-plots if you like, to have more pseudo-replicates if you like. But otherwise I think it's very, very difficult with such long-growing, slow-growing plants to just repeat the same experiment over and over again.
Yeah, yeah, indeed. That's a central question also for us that we are really struggling around.
In our controlled growth conditions, if we look at, for instance, growth cessation and so on, we would repeat the same experiment 2 or 3 times maybe. Just to say that, the experiment was repeated with a very similar result, but we wouldn't calculate any statistics or anything like that.
And I see that you have several yellow sticky traps. You use that to? Are you monitoring the presence of pests?
Yeah.
And what did you see? Because this is indeed a very suitable climate for outbreak of pests. But how is it going up to now?
So far we have not had any problem. We have a little bit of flies, you can see. But I mean, we grow them in one cycle, 2 to 2.5 months, and then we clean out everything, start a new cycle. We don't mix old and young, so they're all at the same stage. So that keeps the problem down.
And you say that this is like a second version of the facility that there is in Gand.
Yeah.
Which
this system is different.
Yeah.
So,
which improvements? I mean, the one you can share, because maybe some are like patented.
but this, I think, is a major change. The system for in Gand they have bands, like belts that are moving. They had problems with them. The resistance was too high sometimes for doing this. These chains but they have these rollers.
Yeah.
I think that is the major difference actually with the propulsion system.
And I see that
This is a bit larger system than in Gand also.
I also see that sometimes the movement are a bit like you say hard. Do you think that this is going to affect a bit the roots?
Well, it affects the growth of the plant. It kind of simulates wind movement. So these plants, if we grow them out in the conventional greenhouse, we have to support them with sticks. Otherwise they fall over. But not here.
Okay.
So they compensate for this movement by getting a stronger stem.
Yeah.
Which makes them, so this is in a way, if you like, more natural than having them supported in the greenhouse.
Yeah. And I see that in IPK they opted for the gantry version, so the plants don't move and they use wind on them.
Yeah.
And in Wageningen there is like instead of a compromise between this system and the one in IPK. So we have one side in which we will have a conveyor belt and one other side with a gantry to compare the. What do you think about?
I really don't know how big the difference will be.
And when we were in Gand, indeed, we had this discussion with the people of WIWAM, and their advice was that in our case, since we would probably deal with very old and big plants, the cost of the conveyor belts or also will be too high to be able to hold such a large tree. So.
Okay, so you're going for such big trees.
Well, that's the initial idea. I'm still partially convinced that we should find a way to use smaller trees and try to see if there are correlations between some traits of the young with the performance in field of the old one. That will be much more pragmatical, but unfortunately, indeed, like there is also the fact that the hazelnut takes 5 years to become reproductive. So, yeah, a reproductive plant is different from a non-reproductive plant.
We discussed our watering cycle. We are not doing any imaging at this time. That happens later in the day. Then we have the infrared camera in the first cabin, and we have RGB cameras in the second one here. So we have top camera, side camera, and the bottom side camera to just image the stem.
Are you developing a 3D model, like using functional structural modeling?
We are trying to see if we can use the RGB pictures to create some kind of 3D model. Or at least getting a feeling for the leaf area index.
Yeah.
We have a project right now, actually, with some students trying to design models for getting very good shoot biomass estimates just based on the images.
Yeah. And okay, you just mentioned, for example, like a difference between greenhouse and this kind of system, talking about the stem. But what is the main difference that you see when plants are growing in this control environment versus outdoor? What is the gap that is still not, sometimes I read about specific leaf area. There was a nice paper of Porter that talks about the main traits that still are like differing between control environments and open field.
I shouldn't say that we have systematically looked at this, actually. Really trying to compare the phenotypes between the field-grown plants and under these very controlled conditions. We haven't done that yet.
Okay.
There are, of course, many more different stresses that they are subjected to outdoors. So here they get very, very uniform temperature conditions, water conditions, light conditions, pest conditions.
Yeah.
There are so many more different stresses that they are exposed to outside that we will never find here.
And are you studying also fluctuating environment within this facility?
No.
Yeah. So you're doing for the so-called square wave, not light on, light down, not like. Okay.
As I said, our main goal is to decipher the role of the genetics here, of the genes, under very controlled conditions. So we want to change as few parameters as possible.
Yeah.
Just change the genetic composition. And then see what the effect has.
And are these sexually mature or not yet?
They will be sexually mature in 10 to 15 years.
Okay. So yeah, the only one that is quickly is Birch.
If you treat it with carbon dioxide, you can get the birch to flower in a year. Otherwise, I think it's also like 4 or 5 years as with Corylus.
As far as you know, do you think, because we know that they indeed like are from the same family, do you know if Corylus has the same possibility?
I don't know if anyone has tried.
That could be interesting to see, because that could be again, something, a plot twist for our project. Yeah. So, like the facility, when was open, like when you put inside the first plants?
About a year ago.
Okay, and currently these these experiments are leading to publications or are internal tests?
No, this is a real experiment. This is the first real experiment we are doing right now. That will hopefully lead to publications. up until now we have been running various tests to try to get the conditions right. Well, this is the first real experiment, and it's a full experiment, right? This is maximum capacity for larger trees.
And exactly the research questions for this experiment?
Identify genes that are controlling growth rates mainly,
Okay.
but also to some extent wood qualities. You can see that there are some trees that are really growing faster than the others here.
Yeah, indeed, there are some that are very small also, I see, yeah.
You see this green dot, that is the control plant. They're called T89.
Sorry, the?
It's a control plant.
Control, okay.
Non-transforming. The others are various combinations of genes that we have identified from other experiments as potential regulators of growth.
Yeah.
You can indeed see that P89s are among the shortest ones here.
Yeah.
So we have various.
And so how many experimental units per cultivar do you have? Like 6, 7?
I think we have 12 replicates or so.
Mm-hmm.
The number of different constructs is maybe...
Yeah, maybe 30 then. So probably between 20 and 30.
Something like that.
Yeah.
Maybe it's up to 16 replicates.
Okay.
Something like that. 16 replicates constructs. Or actually, we have several different transgenic lines in each construct. So maybe we have 3 different transgenic lines, and then we have maybe 8 different constructs.
So if you could have if you have our goal, and if you can think a bit differently than how we think, and you have resources as we do, and
So what is your goal exactly?
yeah. So our goal is to understand where Ferrero can go and plant new varieties faster than now they are currently doing. So, of course, now if they want to expand their cultivation in South Africa, for example, they have to go in South Africa, plant several experimental plots, see how the climate behaves through a number of years, and then take their decision. It is good, it is not good. Instead, there is a strong interest from the management to make this process faster, and the initial idea is that with a controlled environment, with a large facility that can simulate climate, we could be able to make the process faster. So let's say that we do the so-called envirotyping, so we characterize a climate, and we create a representative climate of South Africa, but can also be Australia, and then we expose the varieties of interest to this climate within the controlled environment. So probably we will have to build different chambers or go step by step. So this year we will test South Africa, next year we will test Chile.
It sounds very challenging to me.
Also to me. We agree, we are common
because
ground.
There are so many different variables that will occur over the year, right?
Mm-hmm.
That will be so different in the different locations. And maybe you don't have a control over all these variables. I mean, you need to know. I mean, the easy ones is how much light are they getting, how are the temperatures fluctuating over the year. But the hard ones is how much is the water fluctuating over the year, for instance. What about the nutrient or the soil types or the nutrient composition of the soils?
Yeah.
How about exposure to pests? Things like this.
Yeah.
All of that you get when you make a real field trial at the right location.
Yeah.
But simulate all that. That's a challenge. I mean, it's all about, you might reduce your risk a bit, but would you reduce it enough that you would dare to not make a field trial at all?
So
that's, I don't know.
And indeed, I believe that maybe like in the future we will have to make our goal more realistic. That's also what I'm trying to. That's what I believe in. I don't believe entirely that we will be able to use this system in the way that was originally envisioned. But I still believe that this tool can be very useful. For example, to developing models, crop models. There is only one recently developed hazelnut crop model. So a climate chamber, we could use that, for example, for improving the calibration or understand better like the sub-models of the overall model.
Sure.
So
yeah, that you can do if you want to change just the parameter at the time and
yeah.
see how that affects.
Because in a way that is what you're doing also here, right?
You can easily change the day length compositions. You can change, you have very good control of the watering status, nutrient status. To some extent, you can change light qualities, temperatures to some extent. So these things you can do in a setup like this, sure. If you want to say calibrate your models in that sense. But yeah, if you would really be able to replace the need of field trials, I doubt it.
Yeah, but indeed, let's doubt and let's say that is indeed like let's say that is it's not possible. How about complement the field trials and create and find a way to use the chamber and field trials to cut the time necessary for the management to take a decision. So instead of needing 10 years to decide to go in a location, thanks to the combination of the chamber and the field trials, we could go for 5 years. Do you think that it's possible to make these technologies, open field technologies and chamber technologies synergize?
Maybe. I mean, maybe you can look historically and because I mean one thing that you have is also that you have a great variation between years, right? Mm-hmm. And if you want to really invest in planting a big perennial crop, you want to make sure that it's also stable over the years, right? So, but maybe if you look historically, you can go back and look at different previous differences in weather conditions and so on, and try to simulate this in order to get the feeling for how resilient your trees are to this type of historical changes that has occurred at a certain site. Maybe that could help you to reduce the need of multi-year field trials. But again, I mean, it's difficult to simulate a lot of, there are so many different things that can happen in the field, right? I mean, you have pests coming and going.
And that's environment, and that's impossible. Yeah.
It's almost impossible to mimic that, right? So you would have somehow I have to convince the person that's going to plant this big crop that, well, okay, we haven't really tested for this, but we think that they will also tolerate a new breakout of a fungi or insect or something like that.
No, indeed. Like what I'm trying to push is that the facility we are building, more than an actual copy of an environment, will be a proxy, and we can, let's say, asymptotically get as close as possible, but we will never match it. because there is a so-called terroir of allocation that is not necessarily a magic concept, but it has actually the unique combination of microscopic environmental features.
Absolutely.
About about the control of CO2.
We don't control.
You don't control, okay.
We have quite a large turnover
Mm-hmm.
of the air, so we maintain close to outside
Yeah.
CO2 levels.
Okay. And now about the presence of air contaminants, because this is a facility made of wood, for example, and sometimes, for example, plywood. In this case, is not, but there are some glues, some materials that create artifacts.
We haven't seen.
Okay.
Haven't seen that. And that you can have in all types of growth chambers. I mean, you can have plastics. And
Yes.
You can have
Yes.
lots of stuff. I think it's hard to predict actually. And it's indeed maybe a bit unusual to have a facility built completely in wood. It creates a nice environment also. It buffers a little bit.
Mm-hmm.
So, yeah. No, but it seems to work okay. They are definitely very healthy, these trees. More healthy than they are in the greenhouses. We have very, very little variation in growth between replicants.
So, and apart from measuring like the growth and the wood quality and the wood formation that you mentioned, what other questions are you trying to answer using the RGB cameras?
Yeah, as I said, we are looking into to what extent we can get a kind of 3D model over the plant.
Yeah.
I mean, we want to try to estimate the stem and shoot biomass based on the orbital smoothing. Also look a bit at the leaf area.
Yeah.
But we haven't done so much work on the analysis side yet.
Okay.
There are some projects ongoing for this now.
Yeah.
So right now it's a lot of focus on just looking at the growth rates, looking at the height growth, looking at the diameter growth.
Yeah.
To some extent also looking at the water consumption. We have not really started to analyze the infrared data yet either. It's just some kind of proxy for drought stress.
Yeah. Because indeed, with the RGB you get because why don't you use a grayscale image? Because with RGB you also get some information about the pigments. Are you already doing or thinking to do something on that?
We might.
Yeah.
Sure, I mean, variation in pigmentation is also of interest.
Yeah. And are you using PIPPA? How did you manage to compare that with the, for example, PHES, PHES, developed by INRA or?
No, we have not done any comparisons like that.
And how is it PIPPA is going?
I think it's being developed all the time, right? I mean, one of the reasons we chose this was that we wanted to, as I said, compare experiences and scripts and so on with VIW. VIW used their platform for maize, and that was kind of the closest plant that we could think of in terms of height and size and so on compared to our trees. But I would say that we still have very little, relatively little experience on the image analysis side. This is something we will have to develop more. I cannot really say that we have compared a lot of the different systems. We do very basic analysis right now.
And you have thermal cameras?
Infrared
Infrared
cameras.
cameras? You have fluke FLIR cameras or something else?
Maybe it's a FLIR. It's the one that SMO is offering.
Okay.
For the WIWAM system.
Yeah. And it's like an imaging?
Yes.
Or a point measurement? Like
It's an image.
okay.
We are right now trying to see how we can make that more quantitative.
Yeah. And so that is an information you're gathering because, again, you want to do some studies also on the water use efficiency. So you want information on the stomata and these kind of things? Okay.
Not something we are doing right now, but we want to have that possibility also.
Yes. Okay, can you do like also some indeed frost experiments in this facility? No. Do you study here dormancy or bud induction?
We haven't done yet, but we can, of course. Since we have no outside light, we can easily change the day length here.
Mm-hmm.
And as I said, we can also affect end of day and end of night far red ratios if we like. But for sure we could do, and we probably will do growth cessation and bud set experiments here also. The thing is that we have to do it with the whole platform at once then, right? So it will be one big experiment.
Yeah. But because the temperature has also a role in that, but you will not.
For Poplar, it doesn't have a lot of importance for the growth cessation.
Okay.
Maybe for the speed of bud development.
Mm-hmm.
But yeah, we can lower the temperature somewhat. We have a bit lower night temperatures now, and we could lower the temperatures if we wanted to a little bit more, but not a lot. But as I said, for the growth cessation, it's definitely the day length which is the critical thing for Poplar.
Okay.
But if you want to, induce cold hardiness and then later if you want to release the dormancy, then of course you need to cold treat them. And then I will move them into a cold chamber.
And then you bring them back?
Yeah.
Okay.
I see no reason why you would need to do that on the platform.
Yeah,
because they are unresponsive to any environmental conditions at that case. So the only thing that matters is actually
yeah. Okay. The cost of the capital cost, the initial cost of setting up this facility.
It was about 1.2 million euros or something like that.
Mm-hmm.
For the system, I haven't really calculated. I don't really know what the cost of the building was, but we pay rent for that.
Yeah.
The system, as with everything related, was about the light, about more than €1 million.
Yeah, the lights were the most, or not?
The lights are not that expensive.
Okay.
It's this system which costs.
And the running costs, because there is like a lot of electricity.
It's not so much for the LED lamps. It's not a lot of electricity actually. It's not so high. Depreciation of investment is the major cost.
Okay.